Welcome to the first lecture of your Unit 2 in Honors Biology. In Unit 2, we'll be talking all about the cells, the organelles that make them up, the processes that they carry out, and why they are as tiny as they are. However, in this first lecture, what we're going to talk about is about cell history. So you're going to get a mini history lesson. I will also throw in, just at the end of it, a little bit about microscopes and the different types that are most commonly used in biology. In order to start talking about cell history, we're going to start with a guy named Robert Hooke. Robert Hooke is known for a lot of different things, but for us, what we're most concerned about is the time period that he was studying cork. And I'm talking about the cork that makes up cork boards that you might have in your room, cork stoppers that you put in wine bottles. That's the cork I'm talking about. And what he did is he took a very, very thin section of the cork and he put it underneath a microscope and then looked through the lens and saw what he saw. What he saw was, and what he named, are what we're interested in. When he looked through the microscope, he saw tiny, empty chambers. Tiny little rooms is basically what he saw. And at this point in time, he was connected with a monastery. And the monks that lived at the monastery lived in cells. Those tiny empty chambers, he coined the term cells. And so that is what we use today. But he got it from the rooms that monks live in a monastery. The next guy that came along was Anton van Leeuwenhoek. He was a Dutchman. And he was actually known as the first microbiologist. He's considered to be the father of microbiology. One thing that he is credited for a lot is improving the microscope. He used different techniques in order to make the glass in a microscope, the lenses, thinner and easier to make. And that actually made the microscope a little bit easier to use. Once he made these improvements to the microscope, he studied anything and everything he could. The one that we're most concerned about, the item that he chose to study, was actually pond water. Before this, everybody thought water was just water. It didn't have anything else in it. But when he looked at pond water through the microscope, he saw that it was teeming with life. One drop of pond water had just so many different things in it that it wasn't just water. And through his improvements through a microscope, he could definitely see this. The third guy, and he's got my favorite name, is Matthias Schleiden. Schleiden was a botanist, meaning he studied plants. Once you made the improvements to the microscope, it opened the world up to a lot of different things, and a lot of different scientists started studying more and more about their fields of choice than ever before, and they could see a little bit farther and a little bit deeper. With Matthias, he eventually came up with a portion of the cell theory, and what he stated is that all plants were made of cells. On the flip side of that, you have Theodor Schwann. Theodor was a zoologist. So on the flip side, he studied animals where his buddy Matthias studied botany or plants. What he saw when he went a little bit deeper and looked at different things underneath the microscope is that all animals were made of cells. The last guy that I want you to know about is Rudolf Virchow. He is actually the one that's been accredited with coming up with the first cell theory, actually putting all of the bits and pieces together. However, some of his things that he came up with no longer are relevant to us anymore. So the one thing that he was right about, and the one thing I want you to know, is that he stated that all new cells can only be produced from the division of existing cells. Now, if we take the last three guys and we put it all together into something coherent, we come up with the cell theory. This is the modern cell theory. There are 3 parts to the cell theory. First one takes what Matthias and Theodore came up with and puts it all together. Since they studied plants and animals respectively, then the very first statement of the cell theory says that all living things are composed of cells. The second part of the cell theory states that cells are the basic units of structure and function in living things. The third statement of the cell theory is all from our last guy, Rudolf Virchow. New cells are produced from existing cells. 2 of our units that we will talk about this semester will be solely based around this part of the theory, where our new cells come from, whether it's through mitosis or meiosis, and eventually you guys will know exactly what all that means. Now, the last part of this lecture that I wanted to do is just to give you an introduction to the microscope, or the different microscopes that you can come across. The very first one, the one that you guys are going to be familiar with, are light microscopes. These are the ones we'll use in class. These are the ones that you'll use if you take anatomy with me. It's what you would use mostly in college, unless you do research or something like that, and then you'll come across the next 2 that I will talk about. However, with light microscopes, these use light to view small objects. However, these are very limited in detail and resolution. The second big type of microscope that you could come across if you do research, and this is the one I used a lot in my research that I did in college with my professor that I worked for, is a transmission electron microscope, otherwise known as a TEM. These use a beam of electrons aimed at a sample to study cell structures and protein molecules. In order to use this microscope, samples must be preserved in some way, and samples must be sliced ultra thin. And I'm talking really, really thin. Otherwise, this type of microscope will not work. A lot of times the images that you get with this are considered to be 1D images, just because of this ultra thin sample that you would have. The last one is the one that I think is the coolest. This one I absolutely love because the pictures that you can get from this microscope is just astounding sometimes. The last one that we're going to talk about is the scanning electron microscope. And you guys can finish spelling that out. These use a pencil-like beam of electrons to scan the surface of a sample to produce a 3D image. In order for you to use this, samples must be preserved. You'll find that with most any microscope that you'll use. But samples can be scanned, and this is where it's really different from a transmission electron microscope, can be scanned whole. They don't have to be ultra, ultra thin. It doesn't have to be a small, small sample. It can be scanned whole, because the main thing with this one is that you're scanning usually the surface structure of a bacteria or a dust mite or whatever it is you are looking at. You scan it and you can look at it as a whole. That concludes this lecture. Bring your notes to class, and I will see you there.